My name is Arti Ahluwalia and I'm a professor of biomedical engineering. I'm also the director of the research centre, Centro E Piaggio. It's a multidisciplinary research centre. So what we're trying to do is to use the human body as an inspiration for making better robots, better machines, and also for improving the quality of life. Very inspirational to work here. I can see the tower from my window. I think Pisa, like the rest of Italy, is really the birthplace of European culture and of a rational scientific approach, a scientific way of thinking. And that is a great inspiration for all of us, because we really do, rather than being inspired by ideologies or beliefs, we're inspired by evidence and rationality.
Obama told a few years ago that we are going to understand the galaxies and atoms, but nobody knows anything about the brain. That's what fascinates me. It's a mystery and it's a challenge, and I like challenges.
The aim of the DataBrain Project is to use deep imaging techniques like confocal and two-photon microscopy to image clarified brains and thereby extract the structural morphometric information of neuron architecture in its native 3D environment, and then use that information to 3D print the neurons. In that way, zooming into the brain architecture, we can see how, for example, diseases or disorders such as autism or Parkinson's can alter the structure and therefore the functional connections.
The structure of the brain is strongly connected with the function of the brain, so giving to the scientific community a high-fidelity map of the brain can help to understand how the brain works. To digitalize a map of the brain, we need to acquire the brain at cellular resolution and deep in the tissue. But the brain is full of lipids, and we need the X-Clarity machine to clarify the brain and to acquire the best contrasted images.
Tissue clearing methodology and the electrophoretic tissue clearing machine that we're using by Logos has actually been fundamental to the DataBrain project, because without being able to clear tissues of the lipids, we wouldn't be able to image deep into brains, and so we wouldn't be able to extract that structural information that is so important to understand the function of mammalian brains.
My algorithm works on the pixel intensity of the brain, and it automatically reconstructs single neurons. So we can have further information about the neuron volume, for example, dendrite thickness.
I think it's been a huge advantage, and it's great leap forward in imaging, not just of brain tissue, but also of other types of tissue, because we can now clarify different organs. Mini organs as well can now be clarified and therefore visualized in depth without having to resort to techniques that break down the tissue, and therefore we somehow lose the structure while we're actually trying to investigate it.